Good evening, everyone. Welcome to Botany Insider, another free session on our YouTube channel. We are going to discuss about unit number 3, that is nucleic acids and replication concept, which is related to your molecular biology, right? So, we have already covered few topics of unit number 1 and unit number 2 in our previous 4 classes. So, this is the fifth class for you all. We'll be having regular sessions every single day till our main examination, that is till 26th of December, right? Depending on our examination day, we'll be discussing or we'll be having the classes accordingly, be it on 26th, 27th, or 28th. So we'll have the classes accordingly, right? Let's begin with the class now. So this is the fifth session. If you have not watched the previous one, make sure that there's a complete playlist for the December 2023 paper. You can use that and get yourself a step ahead in the preparation. So if any of you is targeting the June 2024 examination, we have a full course ongoing for this particular examination with it's a all-in-one course with every single unit being dealt with educators. You have part A also dealt in the same. Hard copy notes will be delivered to you at your doorstep, right? In all the recordings, class PDFs, everything will be available to you. Daily practice questions are dealt in the class. Then we have PYQ sessions, weekly test, regular doubt session, every single thing in this one single course. So it's a all-in-one course for you all. Those of you who wish to enroll, the link for the same is in the description box. Right now, we also have a mock test series going on for the upcoming examination for the December 2023. If any of you who wish to enroll in the mock test, because yes, obviously the practicing out of the mock test is very important, or practicing out the questions is very important during this last time. So links for all the mock tests, all the courses, our application is in the description box. You can download it from both the Android as well as iOS, right? Hope it's clear. Let's begin talking about unit number 3, that is molecular biology. First thing first, it's a very factual unit. You should have all the facts very much clear in your head to get a very good hold over this particular topic. We majorly get statement wise questions from here. We get questions in match the following majority of the time. So prepare your unit in the similar pattern, right? First is the DNA replication concept, in which you had to learn about what is DNA replication, the repair and recombination, the various enzymes involved, etc. Then you have the second concept of RNA synthesis. So in this we learn about that what transcription is, what are the various factors for the prokaryotes and for the eukaryotes individually, depending on what is asked in the examination. If you analyze, so there is no bifurcation as such, or there is no favoritism as such. For both prokaryotes and eukaryotes we have seen questions, so do not skip any of them. Then you have protein synthesis, then you have the control of the gene expression as well. If we have a very clear picture of what is there in this entire unit, so you have the discussion of the central dogma of biology. So, what is central dogma of biology all about? Central dogma of biology says you have the DNA getting converted into your RNA, RNA getting converted into your proteins, right? Now, the if you want to increase the copy number of DNA, you have the concept of replication. Then DNA converting into RNA, you have the concept of transcription. For RNA getting converted into protein, you have the process of translation, right? If by chance there is any kind of problem in our DNA content, any kind of replication error is there, we have various mechanisms of DNA repair, right? Then the DNA copy number after getting increased. So this DNA replication occurs in the S phase. We have discussed about this in our previous sessions, right? In the S phase, which is your synthetic phase in your interphase of cell cycle. Over here, you have your MCM helicase required. This MCM helicase is provided via or from your G1. It is synthesized in the G1 cycle itself, right? Then the DNA getting converted into RNA with the help of transcription. What is transcription? It's a process in which the double stranded DNA gets converted into single stranded RNA. RNA then gets converted into the protein, but there's a important step in the middle, that is DNA getting converted into RNA. It is not in the format of your main RNA, it is in the format of your primary RNA. That is, your RNA should be, it is not in the mature format, so it need to be converted into the mature RNA. So for that we have the process of RNA processing. In the RNA processing, there are three sub processes which you have to remember. First one is your 5' capping, you have 3' tailing, and you also have the concept of splicing. Why is splicing done? So splicing is done because you have the introns, which are the non-coding part in our DNA, right? Non-coding part in our RNA. So these introns need to be removed off. They are of two major types. One is your group 1 introns, and then you have the group 2 introns, right? Group 1 and group 2 introns. So group 1 intron and group 2 introns are based upon the number of the trans esterification reactions that we have, right? So in group 1, you have one trans esterification. In group 2, you have two trans esterification reaction. Then your main mature RNA gets converted into the protein with the help of your translation concept, right? With the help of the mechanism of translation. Over here, you have the final formation of the protein, right? So you have to focus upon a number of topics in this entire unit. One is the DNA repair. First is DNA replication, then you have DNA repair, then you have transcription, then you have processing, then you have translation. And after both of these, you have post over here you have post transcriptional modification, which includes your RNA processing. Over here you have post translational modifications as well, right? So keep a note of all of these things. Very, very important. This is the entire gist of your complete unit, right? Then, if I look into my first segment, that is the nucleic acid. So, nucleic acid are made up of what? They are made up of nucleotides, right? What are nucleotides? They are again made up of nucleosides and your phosphate group, right? Nucleosides are made up of nitrogenous bases and your sugars. Now, nitrogenous bases can be your purines, they can be your pyrimidines as well. In purine, you might have adenine or you might have guanine. Both of these are a part of the same, right? Then you have the pyrimidines. In the pyrimidines, you have cytosine, uracil, and your thymine being there. So now the sugar can be either deoxyribose sugar or it can be an oxyribose sugar. Remember about all these segments. It's very, very important, right? You can get basic level questions from here as well. Then you have the pyrimidine structure and then you have the purine structure as well. If you look into the biosynthesis of all of these, every year we can expect a question from here, and in 2022 also there was a question asked from this segment. So this is how the structure of our pyrimidine looks like. And in the structure of the pyrimidine, first nitrogen
sixth carbon, fifth carbon, and your fourth carbon. These are formed, or these are via your aspartate. Then you have second carbon from your CO2. Third nitrogen is from your glutamine. Very important. Remember about these. Then you have the biosynthesis of your purines as well. So in this, your first nitrogen is from aspartate. Second and eighth carbon is from N10, formyl tetrahydrofluorate. Third and ninth nitrogen is from glutamine. Fourth, fifth, and seventh nitrogen. Fourth, fifth carbon and seventh nitrogen is from glycine. Sixth carbon is from CO2. That is the carbonate. Remember all these things, very important, and they have been asked previously in the examination also. Right? Remember this. Then you have the various forms of DNA. So we have A form, B form, and the Z form of DNA. A form of DNA, B form of DNA, and the Z form. So A form, helical sense is right-handed. B form, helical sense is right-handed, and for the Z form, it is left-handed. Then the diameter over here is 26 angstrom. Over here it is 20 angstrom. Over here it is 18 angstrom. Base pairs per helical turn is 11, 10.5. Well, you can easily get a question from this particular table. Okay, so remember about this. Then you have helix rise per base pair, 2.6 angstrom, 3.4 and 3.7 angstrom. Then over here you have sugar pucker conformation. So it is 3 dash endo. So C3 endo over here in the P form, C2 endo, and over here you have C2 endo for pyrimidine and C3 endo for your purines. Remember about it. Glycoside bond formation in case of right form anti. In the B form also it is anti, and in the Z form it is anti for pyrimidines and syn for the purines. Then you have the major grooves. Over here it is narrow and deep. Over here it is wide and intermediate depth. Over here it is shallow and flat. In the minor groove, in the A form it is shallow and wide. Over here it is narrow and intermediate. Over here it is deep and narrow. Very, very important. Okay. Very important. Very, very scoring. You can get match the following questions. You can get statement wise question. Any type of question can be framed from here. So remember all these segments. I have made this table with a lot of information in it. So remember this, it will help you out, right? Go through this, then we'll proceed further. Good evening. Right, this is about the A, B, and Z form of your DNAs. I'm taking up these classes so that it might help you in the upcoming examination. But maybe students are not able to come in the live session, but many of you watch the recorded part. So make sure that you are going through all the recorded parts every single day, right? Then we have the various modes of the DNA replication. So, which are the various modes of DNA replication? So, you can have semi conservative, you can have conservative, or you can have dispersive mode of the DNA replication. So, semi conservative, conservative, or your dispersive mode. Semi conservative is two strands of DNA unwind from each other and each act on the template for the synthesis of new complementary strands. So, semi conservative is when you have the parental strand, and in the next generation, you see exactly half of the copy is given to one of the parent, one of the daughter, and the other is synthesized new. Conservative, from the first generation to the next one is the exact copy and the other one is synthesized completely new. In the third condition, dispersive, over here you have the both the parents having few both the daughter cells having few stretches of the parental in different dispersive mode, but this does not exist in the biological system. So remember about this semi conservative. This is the main correct mode of our replication. Over here it is conservative, in which the parent and one of the daughter is exactly similar and the other one is exactly newly formed, which is also not there in the biological system. Dispersive is the one in which you have the parent, and in the next generation you can see dispersion of the parental. See over here, this is the red color DNA, and in the next generation you can see orange color with red color dispersed at very unequal fragments in the parent, in the daughter generation, right? So second, conservative and dispersive, both of these does not exist in the semi conserv in the biological system, but the semi conservative is the one which is the correct mode of the replication that is found in the biological systems, right? Remember about this. See over here, you have the, when we look into the DNA replication going on, so we have a number of enzymes working in. For example, first one is helicase. So what's the function of helicase? It helps in opening up your helix. It helps in making the double strand into a single strand in this particular orientation. Then you have topoisomerase. So topoisomerase helps in what? It helps in removal of supercoiling. So supercoiling gets removed in with the help of topoisomerase. Then you have the third one, which is your single stranded binding protein. So what's the functionality of single stranded binding protein? They help in maintaining the DNA's single strand, because otherwise our parental strand have the capacity, because they're complementary to each other, to get binded or to get wounded into the double strand very quickly. Then you have DNA polymerases. So DNA polymerases have the functionality of adding on your nucleotides, adding your NTPs in the progression. Then what else do we have? We have, yeah, these are the major ones. In addition to that, we also have helicase is done, topoisomerase, single stranded binding protein. Primase enzyme is also there, which helps in the formation of the primer, for which, upon which our DNA polymerase can actually start working on, because your DNA polymerase cannot initiate the work itself. It requires a primer. So a primase enzyme is required. DNA polymerase, based upon your prokaryotes and based upon your eukaryotes, you will see a change, or you will see a difference in the prokaryotes and the eukaryotes with the polymerases being there. For example, in case of your prokaryotes, you have DNA polymerase 1, 2, 3, 4, 5. And in case of your eukaryotes, you have alpha, beta, gamma, theta, delta, epsilon. So all of them have a different functionality. Remember, make sure that you are revising all these things properly, because I'm just giving you a gist of the concept. You need to revise up the things very nicely, right? It will help you in scoring good in the upcoming examination. So remember about this, right? Then you have one question over here. So this was June 2022 question, sorry, June 2021 question. Question is, you're given this figure: replication of the DNA beginning from this origin of replication of the genome of the chromosome of a newly identified bacterium having a double stranded circular DNA genome is shown to you. Characterization of the DNA polymerases responsible for the genome replication showed that the DNA synthesis occurred in 5 to 3 direction, right? And it depends on the presence of the primer, as in case of E. coli. Polarities of the DNA as shown 5 to 3. This is also 5 to 3. So it is asking us, what does the model A depict? What is it actually depicting? So is it a unidirectional mode or is it a bidirectional one? First thing first, it's a unidirectional. Why? Because even if it is showcasing in the opposite directions, but both of the directions are same, the replication directions are same, so this is not bidirectional, it is unidirectional. Now if it is, this is your origin of replication, so C will be synthesized first and then B and then A. That's what we learn in our main unidirectional mode of replication, right? This is a very basic question that was asked in the examination, where you were asked about that what is the mode of replication, and second thing that was asked was related to that which particular Okazaki fragment will be synthesized first. So Okazaki fragments, first thing, they are synthesized on the lagging strand, right? And the normal leading strand proceed in the normal direction. Remember this. So because origin is here, you have to observe the directionality. Both of them is showcasing the same directionality, 5 to 3. If the directionality is similar, that means it will be showcasing your, it is a unidirectional mode, and in the unidirectional mode the Okazaki fragment which is nearest to the to your origin of replication, that will be synthesized first, followed by the other one. So first you'll have C getting synthesized, and then prior, like after that, you'll be having B and then A. So C is the correct answer for this particular question. It was a 4 marker, easy question, interesting question, base level, and a very basic question was asked in the examination, right? I hope this particular question was clear to you, right? If yes, please let me know in the chat section, and then we'll be moving forward. Okay, just check. Very easy question and very scoring question also. Many students thought that it was a bi-directional one. No, it's a unidirectional one, right? So don't get confused in this particular thing. Simple question. Yeah, perfect. So these were the things that I had to discuss. See over here, one more question. The long DNA strand depicted below is serving as a template for the lagging strand DNA synthesis. It is asking the short line represents the newly synthesized Okazaki fragments. Which of the position will be, or where will you see the DNA primase acting first? So over here, B, C, or T. It's again a logical question. Why will it DNA primase act over here, or at B, or at over here? Because it need to, like Okazaki fragment need to be initiated. So for that, our DNA primase need to work here, right? So that my RNA DNA polymerase can start working in this scenario. So logical question again, you will be having answer as option number D. There's nothing to think about also in this question. If you know about what Okazaki fragments are and how does your primers work, you'll be easily able to mark your answer. And I've just told you about the functionality of primer. So it's not a difficult question at all. D is the correct answer for this particular question. It was a 2 marker that was asked in part B. So remember about this particular thing as well. Right? So these were the questions and the complete concept that I had to take up today. Remaining things we'll do in the next class. Hope all the things are clear. If you're looking for a full course for the June 2024, so we have a full course ongoing for you all. If you wish, you can enroll in the same. You'll be getting hard copy notes delivered to your doorstep. Okay, all the best. Bye, everyone. Take care.